Thank you for being here for the session now. So we are now pleased to welcome Dr. Erika van der Waal from the University of Lausanne, Switzerland. Erika did her PhD in the University of Neuchâtel in collaboration with the University of South Africa and the Louwscope Dame Nature Reserve, sorry if I say this wrong, to study social learning in wild vervet monkeys. She's then joined the University of St. Andrews, UK, to start the Inkau Vervet project in collaboration with the Mawana Game Reserve in South Africa to examine the foraging techniques in social learning. So she's now the director of the Inkau Vervet project, and it's an experimental field site with a study population of over 200 wild vervet monkeys, and she's also a professor at the University of Lausanne. With our groups, they study the evolution of primate social behaviors with the main focus on cultural transmission and cognition. So today, Erika will show us how they used field experiments to eliminate the 3 phases of social learning in vervet monkeys. And I would like to thank you again for joining us today, Erika, and the microphone is yours.
Thanks a lot for this lovely introduction. And yeah, it's my great pleasure to be joining you all online to present some of my research. But before I start, I always want to first acknowledge that all the slides I will present come from collaborative work. So I really like this African proverb: if you want to go fast, go alone. If you want to go far, go together. And so none of my research would have been possible without many people helping me in the field. And I cannot put the names of all the people that were involved, but I hope that with this they can recognize that they are really an essential part of this work. So I've been always really puzzled and fascinated by the diversity and the complexity of human culture. And then I wondered how much of the origin of this culture can be found in non-human animals. And so the building block of culture is what we call social learning, or just learning from others. And actually, when we investigate social learning in many animals, we find that it's very widespread. From insects to great apes, we will find that they learn socially, and even solitary species like this tortoise here, if you give it the choice to follow the foraging route of others, they will do it. So it really seems that actually the default way of learning in animals is learning from others. And so I've been focusing my whole career on vervet monkeys. So why vervets? So they don't all read the French psychology textbook upside down, but they are known to be opportunistic, which means that they engage very easily with human artifacts. They are very curious. They are not endangered. They're even considered pests in South Africa, where I work on them. So we're allowed to capture and to conduct field experiments. Another aspect that is great is that they're semi-terrestrial, so they're very easy to observe in the field, and also, of course, to attract to experimental setup on the ground. And then finally, I think their group structure is really ideal for my kind of research, because average group size is about 30 individuals, which means that one can study multiple groups. Which is always needed when you conduct experiments. You need to have your experimental groups and your control groups. And then another very nice thing of this structure is that male disperse when they reach sexual maturity, but they don't do it only once. They do it repeatedly, so which gives a very big fluidity to the group. And then so that when we test social learning, we can always have related, unrelated group members. And then another aspect that is really nice, but that is not the focus of my talk today, but that you can, of course, ask me question at the end about, is here summarized on an article paper from the Swiss press, these alpha female that dominates males. So actually, we found that in 2/3 of our groups, we had alpha females on the in the groups, which was in contradiction with textbooks saying that in vervet monkeys there is always an alpha male. So I kind of like this power of the females in my species. Where am I studying them? So as said in the introduction, I set up now 13 years ago the Inkavu Vervet project. Inkavu means monkey in Zulu, because we are in the province of KwaZulu-Natal in South Africa. We are working there on private land. And the image you see here at the background is an aerial picture of the field site. You see here a river going through all the different home range. Each home range here of a group is in a different color. As you can see here, the name of the group. So there we have now 7 groups, over 250 monkeys that are followed nearly daily. What is really unique about this field site is having that many groups of primate of the same population. So before we knew a lot about individual differences within a group or population differences in primates, but the neglected level of studies was between group differences. So that was what motivated us to set up this field site, and also to do experiment in the field, which is usually not possible on endangered primates. It makes it a very nice field site to look at social learning, because as you see, groups have a lot of overlap in their home range. So the ecology is very similar, so we can control as much as possible for the ecology, and also genetically, it's one population. We have dispersal across all the groups. So then if we find group differences, then social learning is very likely to be the underlying mechanism. So here I show you the 107 monkeys that participated to the food coloring experiment that I will mention to you later on. So we are all trained at the field site to ID the monkeys facially. And so this really relies on having a large team on site all year long to be able to collect the data and know who is who and who is going where. So I'm going to take you a bit through my journey of social learning experiment. And first I'll start with the first experiment I've done during my PhD, so which was with this 2-action box. And as you can see, from one color end, there was a pulling door and a sliding door on the other one. And there I was really interested in the mechanism of social learning. Can vervets learn how to open a door after observing a conspecific? And so for this, I first brought one box and tried to see who would monopolize it, and then train it on one action with the other action being locked. And so there I had in 3 groups an alpha female monopolizing, and in 3 groups an alpha male monopolizing. And then I looked at what happened, of which side they would manipulate and would they be successful. And when I put everything together, I didn't have very clear results. But then I realized that if I separated with the sex of the demonstrator, then it changed. So here you will see just already the participation of group members. Here we see in the first 3 groups here, participation is really high when it's a female model, and it's very low, lower than 50% when it's a male model. So there is already something going on there. Then we looked at the side of opening. So in black, if they open the same side as the model, and we see that the majority open the same side in the female group, the groups with a female model, and then totally randomly in the group with the male model, which was really surprising. So then we looked at around demonstration, what happened to explain this difference? So first I wondered, oh, maybe actually the males were more aggressive, or bystander didn't dare to be close by to observe. But this is the first 2. So here the number of bystanders, and here the aggression rate. And there was no difference between female and male models. And even here, the most aggressive was the alpha female of the picnic group. But where we had a significant difference was the probability of bystander looking. And here we found that if the female was opening the box, then the majority of group members were having their head orientated towards the box, which was not the case with the males. And I think that was really surprising, really showing the selective attention towards females. So as I said before, male dispersed, female are philopatric. So this task is about resources, and females are the one knowing where to forage in the group and are the core group members. So actually, it does make sense that the group would pay more attention to them. And another aspect that was really interesting was actually were they successful in their first opening. And so here we see that the female model and male model were not important for being successful, and that the two groups where individuals were successful, so in gray and black is the percentage, the monkeys that did manage to open at their first trial, and in white, the one that were not successful. And these two groups were actually groups in contact with humans. So it was the picnic site group and the fishing camp group. And it seems that having human artifact in their home range really made them better manipulator. And so, which also put me, like really triggered my attention to, we should study primates in different habitats with humans around or not, to really see how the difference in the context where they grow up impact their physical cognition. And you'll see at the end of my talk that we started now working also on urban vervets. But actually, my initial questions was to know, can they learn how to open? And actually, I realized that my task wasn't a good one, because the two different doors were on two different colored ends. So I couldn't say if they actually just learn what side to manipulate or how to manipulate it. So then during my postdoc with Andy Whiten, I designed a better box with the same door, a knob that could either be pulled or slide to the side. And there I found that vervets would use the same technique as demonstrated in their groups and this time we really targeted adult female as demonstrator, as we had seen previously that they learned more from females. And then in another task with sanctuary vervets, we even went one step further in with what we call bodily imitation. And there we found with this little tube that models did one of these three techniques. And this was spontaneous. We couldn't train them on one. And then that group members would adopt, and as a majority, the technique they had witnessed to open. So really that there is this attention to how others are solving a task. But so after this experiment, more on the mechanist side, I was really more interested in understanding what we call the social learning bias. So when and from whom individuals learn socially. So I did a modified version of my initial box, as you can see here with the two colors. So this was my PhD student, Axel Bonneau, who conducted this experiment in the field. And I really liked the way the press talked about it. Female monkeys don't trust males even when they're obviously right. So what have we done there? So here you'll see a little video clip. That's the alpha male in one of our group. And you'll see that he opens the black side. So we can control with a remote control. So we unlock when he's coming the black side. And you see that he's very lucky getting 5 pieces of apple out of the box. And so this time we changed the payoff to see, is it really the identity of the monkey or is it the amount of food that is important to learn socially? So what we did is have 3 experimental groups with males getting 5 times more food than the female model, opening the other side, as you will see in a minute. So here is the alpha female of the same group. And so she can open only the white side, and she has only 1 pieces. And then we had 2 control groups where we repeated what we had done before. So having only 1 piece on each side to see if they would, again, copy more the female side of the box. And then we waited that 2/3 of the group had been watching both openings, and then offered the box to other group members and watch at their first opening. And so what did we find? So first in the control group, we found again the significant difference that they would copy more the female side of the box, so confirming our previous result. But then in the experimental group, this disappeared. And so then we looked at what factor could have been affecting this, who didn't copy the female anymore. So we look at age, sex, and rank of the participant. And what we found was that only the sex of the participant was significant. So in control groups, there was no difference at all. Both males and females would copy more the female side of the box. But then in experimental groups, there we saw really a huge difference, with female being highly conservative and copying still the female side of the box, despite the fact that she has a lower payoff, and males showing a greater behavioral flexibility and copying in this context the male model. And so I think this is really interesting and goes well with the ecology of the species, because as I said before, males disperse when they reach sexual maturity and do it multiple times. So for them to adapt to new groups, they might need to change their social learning rules and to pay attention to different group members, whereas females stay always in the group where they were born. And for them, what is really important is the bonding with other females. And so for their own survival and the one of their offspring, to really be with the core of the group is important. So doing like other female promotes maybe this group stability. Then going a bit further, I really wondered what are the pathways of how things are inherited. So when you think of biological evolution, then it's very straightforward with this horizontal transfer from parents to offspring. But when you think of cultural evolution, then knowledge could potentially be transmitted in all directions. So then I did a review of the literature with Andy Whiten to try to really look into primate work. What evidence do we find for which pathway? And what we found was that there seemed to be three main phases of social learning in the development of a primate. And so I'm going to explain them a bit to you now. So the first phase would be really learning from the mother. And so this would be really happening in young infants, from learning from their mother. And there we expect them to learn a complete behavioral repertoire. And the second phase, so this oblique phase, would be of older juveniles, and then learning from other within group individuals. And that would be some selective knowledge and skills that potentially the mother would be lacking. And then the third phase, which is this horizontal transmission, would be learning after dispersal. And so this would be immigrants learning from new group members, and what we expect them to learn is foreign knowledge or skills. And so now I'm going to show you examples of different field experiments that I've done that illustrate well those three phases. So the first experiment here was a grape cleaning experiment. So I just offered sandy grapes to the monkeys. And here I looked at the first 10 grapes eaten by naive infant. Naive infant are always the second name code here. The first one is the mother. And here you see the 4 different techniques we find that they use to eat these grapes. And you can see here that there is quite a good color matching between mother and infants. And more interestingly, if you look at the second graph, here is the percentage of similarity in the technique used of the infants and the others eating at the same time. And so here you see in black how much they ate like the mother, and the white bar like others that were eating also when they first joined those naive infants. And you see that for all of them, they ate more similarly to their mother than to other group members. So really this selective attention to what the mother is doing. Another good example for this vertical social learning of infants, I think it's well illustrated by this picture. So this is a little Porto here sitting on blue corn and eating pink like the rest of his matriline. And so this was a food coloring experiment where we trained 4 groups to eat either pink or blue corn by adding a bitter taste in one of the color. We did the training while the female were pregnant, and then once the infant were over 3 months old, so on age to eat solid food, we conducted experiments where we offered the 2 colors, but without any bitter taste. So it was just the color, the difference. We had 27 babies that year, and 26 of them learned the local preference of their group. So started eating on the group color and like their mother. And I think the one exception is really interesting. So it was Roycat, the son of Recipite, which was the lowest ranking female in our largest group that was over 50 individuals. And then the little Roycat started eating the same color as his mother. So the mother had been waiting for multiple hours and then grabbed a bit of the previously bad tasting corn and realized it was now good. And then the infant selectively copied its mother and not the other 50 monkeys eating the other color. Then for the second phase, so first with natural focal data, we found a decrease of attention towards the mother. So that was doing a focal while individuals were foraging. And so on this graph, if you look here where they're circled in red, you see that 2 years old juveniles still have a lot of attention. So that's the average look towards their mother, and this huge drop when they are 3 years old. So they really seem to switch their attention. Then from experimental data, what we found with this puzzle bug that I introduced to you before is that most of the successful openers were juveniles. So it seems that juveniles are really keen to learn new things. And then here it's in the three groups, A, B, C, the network, how observation linked to successful opening or not. It's too small, you don't have to go into detail, but what I want to highlight here is who are the successful openers. So the first one here is always the adult female that was our model. Then two of the successful openers were actually the son of the model, but then all these others here in green were unrelated juveniles. So really showing that they pay attention to those adult female models, even if they are not their mother. Then the last example for this oblique transmission was a novel food experiment. So here we offered peanuts in shells to the monkeys, and we wanted to see how they would process them to open. And we found three main techniques: so opening with the teeth from the top or from the middle, or cracking them open with their hands. And then we looked at, with modeling, what were the important factors for a technique to be used or not. And there we found that actually juveniles relied more on the payoff bias than adults. So they would use the technique that was the fastest to open peanuts in the 20 minutes before they started eating, whereas adults really relied more on the copy high-ranking individuals. So they really seem to pay attention towards what dominants are doing, irrespectively if it's a good, efficient technique or not. Then, interestingly, high-ranked and adults were more observed than juveniles. Even if juveniles were eating a lot of peanuts and were potentially more efficient in eating them, it seems that the attention of the group is always towards adult and high-ranked. And this is also really highlighted in the one group where we had infant as an innovator. And then the fact of eating peanut then spread only to other infants and juveniles. And so we did the experiment in winter when there is very little resources, and peanuts being very calorific, we expected it to spread very fast in groups. But if it's an infant who start eating, then the adults are not interested and don't learn. So the reverse vertical or oblique seems not to be happening. And so now we'll move to the horizontal transfers. So here back to the food coloring experiment. So here you see the approximate home range of the four groups I had trained to eat either pink or blue. And so the experiment took place, as I said, when infants were about 3 months old, which was then during the mating season. And what is happening during the mating season in the vervets is that males are moving around. So we had males like this male Elton that was trained in lemon tree, that pink was the good color to eat, that blue was disgusting. And then he dispersed to a group where everyone eats blue. And so for the first time, we could really test the relative importance of individual learning versus social learning in the wild. And I was extremely lucky because I had many male dispersing, as you can see here. and also that they dispersed very nicely from blue to pink and pink to blue, all of them. And you can see here that this experiment wasn't designed to test the male. They were designed for the naive infant, because if they went north, south, or east, west, I would have had no data point, because they would have entered group having the same habit as their origin group. So yeah, this was big luck, I think, in my career. And then we had also 5 males from unknown origin that entered the groups, and so like the infant, naive to corn, and even more to colored corn. So what happened then? So I think the saying, when in Rome, do as Romans do, summarize it well. So here you see a table with the color of origin of the different males. And this is what they did in their new group. So the first column is the first item of corn that they selected in their new group. So you see a tendency to switch, but with still 5 exceptions. But then here, the second column is when the males were not outranked. So when they could really sit in front of the 2 Tupperwares and select and stay for long to eat what they wanted to eat. And we saw that all except one conformed to their new group preference, which was the first evidence of conformity in animals. And then very not long after, a very nice example also in dispersing great tits from Lucy Aplin. And then also another very good example of change of tool use from immigrant females in chimpanzee by Lydia Luntz. Then recently, we've also analyzed with a master's student the long-term data at our field site for sociality. So here the sociality index is the number of affiliative behavior divided by the agonistic plus affiliative behavior. And we did that for 3 years, where we had a lot of natural data over the whole period. And you can see here that we have one group in red here, Angas, who is significantly more affiliative throughout the study period. We also did some simulation to control for the demographics, so to have the same sex ratio, age ratio, and group size. And with this also, the difference stayed significant. And what was interesting is that we had the males that were a whole year as adults in one group, then dispersed to another group and stayed a whole year in another group. So we could compare the sociality when there were before and after dispersal. And there we found that the males that joined this group in red here, Angas, that we nicknamed the love group, became more affiliative than when they were in other groups. So there really seems to be some changes in their behavior, some matching to the sociality of the group, a bit like this is how we do things here, we are nice to each other, or this is how we do things here, and we just aggress each other all the time. And then a last thing for this horizontal transfer is that actually when we wrote the review paper with Andy White, and we had this example with the colored corn of male conforming to their group, and also what was found in the chimpanzees. But we didn't really think of the other way around, that actually in primates, there is often group level culture. And so, not group level, population level culture. And so if there is population level culture, there must be vector of knowledge between the groups. And that actually, who can be those vector of knowledge is the immigrants. And so here in the peanut experiment, we did find that really the immigrants seems to trigger the novel food acquisition. And so here you have the proportion of the group member learning to extract and eat peanut during the first event. And you see like that there is a variation in how many group members are learning it. But then interestingly, if you look over 4 exposure after a successful innovation taking place, how it spread in the group. And so you have 3 groups that look very similar in their curve. And so in all these 3 groups, it was an immigrant male who either innovated in lemon tree or came with the knowledge. What was came from Baidanki and Angas was male that knew how to do it from Nora group that then moved there. And then here, the group in orange had an immigrant that actually innovated eating peanut during the first experiment. But then he was copied only by 2 juveniles, and then he left the group before the second experiment. And then it took time before that the first session, a female started eating, an adult female, but before it was only juvenile. And this is the group I mentioned earlier that had an infant innovator, and there you see that it doesn't spread much to others. And then what was really interesting is that during those peanut experiment, we could really observe muzzle contact being done at much higher rates than in other experiments or in natural context. So here you see an adult female targeting the innovator male, Babelas here, and she is really curious about what he is eating and doing all those muzzle contact, we think, to get olfactory knowledge about this new food type, but she doesn't trust it directly and will really repeatedly smell his mouse, but also the one of others before deciding to eat also some of the peanuts. And so when analyzing those muzzle contact, we could find that they're really targeted towards individuals eating the novel food, and that they're also mainly targeted towards adults and especially immigrants. So I think this goes in this direction that the males could really be vector of knowledge in the vervets. And so that this could be the way that we get population level culture, and also maybe it is one of the reasons wild vervets are so widespread in many different habitats across Africa, is potentially if they have all this knowledge coming from males from all around their group, they can adapt better potentially to change in their landscape due to rural or urbanized area developing where they were initially. So to conclude this part on the 3 phases of social learning in vervets, so I think we found some good evidence for the 3 main phases of social learning. But then I think very interestingly is this last phase of learning from immigrants that we maybe overlooked in our paper, that I really want to investigate further in other behavior domains than just this foraging domain. And so now I'm going to take some time to explain some of the ongoing project of my research group. So recently we brought touchscreen technology to the field. So here it's the second session we did with the NOHA group, and this is Ouagadougou, who has already understood that if he wants to have the reward, he shouldn't be touching the whole screen, but only the blue square. So here, mistake. Yes, the blue, and then he gets again. And so with this, the aim was really to see, can we adapt first this technology to be used in the field, but also to see the cognitive abilities of wild versus captive vervets, and is actually all the tests being done in zoos and lab, is it similar to the cognitive abilities of wild individuals or not? And so here is the summary of the first paper we've done, mainly about participation. And so here we found, as expected, that captive monkeys were participating more than wild ones. Then we looked at who participate in the wild ones, because only not in the captive one. In captive, the group composition was not at all balanced, but in the wild, we found that it was mainly adult female, which is explained by the fact that they are dominant in most of our groups. But then in juveniles, it was the opposite. It was juvenile males participating more. And so this was explained by the fact that subadult males dare to be more on their own. So once the group started moving away of the apparatus, then some subadult males would stay and play and then join the group later, whereas juvenile females would always stay with the core of the group. But what was the most interesting was looking at the performance. So here we use the task with the blue square that I just showed before, but moving on the screen and seeing how many trial they needed to reach a learning criterion. And here we found that there was no difference between the wild and the captive monkeys. So despite the fact that they participated less, the wild ones, when they did focus on the task, they were learning as fast as the wild ones. I think now what I really want to try to do with this technology, investigate more a social cognition task and see if there we see variation between wild and captive monkeys. Then a couple of years ago, I started this ERC project entitled Knowledge Move, where I really focused on immigrants. And for this, we've been putting GPS and accelerometer collars on males to really track their movement, to see if males could be showing some new areas to groups. So here, for example, in one of our group, we suddenly had 4 males coming from a group in the south, and the year after they came in, then suddenly there was a core area of this group in the south that they didn't use before. So this is really the kind of things we want to investigate. And then I also want to see with the touchscreen experiment, can males bring new knowledge to their groups, and when and how, or do they need to have a lot of higher payoff to be copied by others? This is the kind of task that we're busy with. So here you see an aerial map. This part here is our research area. So this is from the logger data, the GPS data of the males. And here we see dispersal events of some of our males. So we have the boundary of the research, of the reserve actually just here. So many males went actually outside and looped for long. And interestingly, it was natal males. So the one dispersing for the first time that went the furthest away. We had the hypothesis that it would be the secondary disperser that would go further. But actually, it seems that the young ones want to like see the world, but then most of them come back and settle in very close by groups. So actually realizing that maybe the good place is not too far from home. So here, just to put I have to, I think, remove the pointer to show you the video. Here you'll see how the GPS points look like on the map. So this is the research area. And you see I need to put the pointer again, I guess, for you to. The research area, and you have a female with a color and a male also in each group. So that's where there are multiple moving together. But here you see, for example, a male dispersing in red and going all around. So we can really see in a dynamic way the movement of everyone. So I think this technology is really amazing to be able to finally know what is happening with migrants, because in primatology we follow the residents, and we sometimes see again after a couple of months or a year a male that we know, but most of the time we have no clue if they died or if they're alive somewhere else. So this is really very, very valuable technology to understand how dispersal works. Then we've used also molecular tools to try to understand better the diet of the monkeys and try to see if males have some preferences of food that they keep or change when they change groups. So here you see on the left data from environmental DNA analysis, and on the right from our observation. And here it's a long list of the 3 species, so all the vegetation eaten by the vervets, and this here at the bottom is the different months of the year. And the larger circle is what they eat mainly. So you see that there is quite some matching, like for the main species that they eat between observation and this molecular method. But what is really nice with eDNA is that we find all these other species that we didn't observe. So either cryptic species, also species very hard to identify, like grass and small bushes, that we can then identify through this method. And it is even more exciting, I think, for the insects. Sorry, my slides seem not to want to change. I changed too. So this is for insects. So it's very hard to know when we're in the field with our binoculars what they're eating. So we have those main 3 classes. But then when we look with eDNA, we find 36 families of insects eaten. And then, of course, to know the species, we would need to collect insects there to really know what they're eating. Because on GeneBank, there are not the species of that area in South Africa already there. And so then we really want to use this method to try to understand if groups have like foraging preferences. And so here we looked at what kind of plants they're eating in summertime, so when there's a lot of choice. And so if we take all group members, there's a huge overlap. Each colored circle is a different group. But then if we look only at adult females, there seems to be some differences with some plant type, mainly fruits here in summer, eaten by some group, but not the others. So we want to understand more if we have these dietary traditions, and then would male adopt them or not. And then very lately, we've been designing with my colleague Benedetta Franschello from the engineering school in Waal in Switzerland, a voice recording color to try to see if males have adapt some of their calls after dispersal. So this will enable us, with the microphone close to the monkey reed, to describe very well their vocal repertoire, especially the soft calls that they have. And then we want to see, we had found in the pilot study vocal convergence in male grunts. So I will make you hear how it sounds like. I hope you could hear. And so these grunts, to record them, it's when males are moving one to another. So it's very hard to be at the good place at the good time. So we have a very small sample size. Then we didn't have the kinship between the males. So we find a kind of vocal convergence, but we couldn't say if actually the males were genetically more related. And so that's why they sounded more like each other. So now we're going to have, with this technology, a good recordings on more individuals of these grunts. And then we also want to quantify the calling efforts of males when barking for ground predators and link it to their rank. And so here I make you a little sound effect. So this is how ground predator barks. Okay. And so what is really exciting about these colors, that we managed to make them really light. And so we have also an auto release option. So now I'll show a little video clip. It goes quite fast. So here we have an individual with the voice recording color, and you'll see it just dropped off here. And now you see the same one without. So we could put them on the monkeys, and then it recorded for 11 days nonstop, and then retrieving them so that they don't have to carry the colors when it's inactive. So this for me was very important, that we could find a way that we don't need to make too many captures. And then to finish, so we've just started a year ago an urban vervet project, also in the same province of KwaZulu-Natal. That's quite close from Durban, on the seaside. And with this, with my colleague Sophia Force from Zurich and Stéphanie Mercier, our coordinator on site, we really want to use the same behavioral data collection as done with the wild vervet, the same experimental setup, and really try to see, did the fact that they adapted to our environment change something in their cognitive abilities, what different behavior also they exhibit. And to finish, I want to highlight an aspect that we don't often think of when we conduct research, is the impact that we have on the land where we are working. So here you see the area of my field site. And so this the research area was rented out of a 10,000 hunting farm. And then having us on site made really the owner think about another way of using their land. And so now, 13 years later, we're in the process of trying to develop the Loziba Wilderness, which would be a community-owned reserve for conservation, and that will really hopefully help uplifting the economy of this very poor area. And with this, I would like to thank you for your attention, and I'm looking forward to your questions.
Thank you, Erika. It was a super nice presentation. Thanks for the videos too. It was nice. Let me just talk about the question. And there is a little gap of time when they ask question on YouTube when we see the question. So I have some, actually. I will start with a very naive question. can you use artificial intelligence to identify your vervet monkeys? Because to me, they look all the same.
So this is something we would like to try. So at the moment, when we use the touchscreen experiments, we have a researcher with a tablet saying which monkey is approaching so that the good game comes up. In my ideal world, in a few years, we'll have it all standalone, automatized in the field, and that any monkey could approach, and then just with facial recognition, it could know who it is. And then it will be, of course, also with solar panel that it recharge automatically. It will be elephant proof and whatsoever. When I discuss this with engineer, it doesn't seem that straightforward. But I think that the vervet's faces are still really different and could have features picked up by machine learning algorithm. But then, yeah, I think we need a lot of picture to train and also to update them regularly, because the juveniles, when I see them every 6 months, they change a lot. So it will still be a lot of work for the field team to update with recent picture a lot. And also they often get new scars and things. So to be sure that it's the good ones, we should really try to go into that direction, but really test how like if it's really working or how many mistakes are being done.
Yeah, it's the same problem with my study system with the grades, where they all look the same. So we have question now coming from the YouTube channel. The first question is from Amanda Frashier. She's asking, do you know what males are doing when they venture off, when they are leaving the group?
What the males are doing?
Yes.
So what is good is that on the GPS collars, we have accelerometer on them. So with this, we can infer their behavior also. So we've been trying to classify, we just had a master student, different behavioral types with specific accelerometer patterns. And it's still very broad category, like moving, resting, feeding, roaming. But with this, we can say that the males are actually moving quite a lot, and when they move faster than with the group. But what they seem to be doing a lot is resting, but we think that actually the problem there is that vigilance goes with resting. And so being a male on its own as a vervet monkey is really dangerous. So they have many predators, and so I think they have to be really careful when they disperse. What we could also see is that they really follow the vegetation paths always, so they seem to stay in as much dense cover as they can. And then what is nice is that using both the GPS data and accelerometer one, we can have an idea when they actually join the group. So there are many uninhabited group around the study population, and so there we see that they stay in the same area and move less, and that they have really a pattern that looks a lot like when they were in their natal group, and then we can infer that they were with other monkeys.
But do they have an advantage doing this, being alone at some point and then coming back?
So I think dispersal is really something that is needed to avoid inbreeding, and is so we see sometimes females actively becoming aggressive towards subadults when they stay for too long in their natal group, and then I guess they are just looking for a suitable population, and it seems that when they move a second time, then they will more go to neighboring groups that they have encountered during between group encounters, and have seen that there are maybe not many males or a lot of females. But their natal dispersal, they seem to be like just leaving and then seeing what they find, which doesn't look very adapted.
Okay, thanks. So we have another question from Catherine Macri. Hi, thanks for your talk. I think it's really interesting that you found out that the wild have the same performances as the captive monkeys. And sometimes it's said that the captive ones are overtrained. Do you plan to try to compare the performances using more complex tasks?
Yes, so after that one, we've done associative learning, reversal learning tasks. There, the problem was that the sample size became really limited on the one that finished up to the end in the time frame of a PhD. And so there we found maybe a little tendency for the captive one to be faster in the associative learning. And here I think it is also a lot about the memory and the fact that the captive ones could be tested at a lot more regular interval, where the wild ones, if they're on the other side of the river, we could not bring the 15-kilo apparatus and take the risk to break it while crossing the river. So they were often things, or there was too much rain or things that made it more gaps in between the trials. But yeah, I think we always have in mind the thing that like captive animals with the free time hypothesis will be more clever because they have nothing else to do. But I think we should also look at the opposite hypothesis of the wild one being constantly challenged by so many things, whereas the captive one don't have to look for food, don't have to defend their home range, don't have to look for predators. So maybe the wiring of their brain is also very different than the wild one. So that's why I think it's really important to compare and find a different paradigm that really test their abilities and to have this comparative approach.
We have another question from Lee George. It's interesting that infants can also teach other infants, even if adults don't learn from them. Do you notice some active behavioral process in which the infants were trying to teach other infants?
I'm sorry, I couldn't hear very well that. So if the infants were active things, and so I didn't understand that part at the end.
I can say this again. Did you notice some active behavioral process in which the infants were trying to teach other infants?
So we didn't find any active teaching, but I think, so generally infants and juveniles are a lot more tolerated by other group members. So it seems that rank has a big impact on who is tolerated by whom in adults, but juveniles can be all together around food without aggression. So I think this can already promote social learning opportunities. And then I still think that the juveniles spend a lot of time with peer age groups. So they will stick a lot with other juveniles of similar age. They play together a lot. And so I do think that they will be in more proximity of other juveniles often. And I guess in the context of the peanut, once the adult they came to the box because we brought the peanuts in the same Tupperware, as you could see, as the corn, that they knew it was really good to eat. So the adults came and smelled and picked up and didn't understand what it was and didn't try to open it. And then the juveniles just stayed for longer. And then at the fourth session, suddenly one infant started to eat and and other juveniles were still around. And I guess adults were also just not interested by coming close to that experiment anymore.
Okay, I have Catherine who asked the question before. She said, thank you for the answer. And I also have a question for you. When the monkeys moving changes their initial choice to match the choice from the group, can they reverse the choice when they go back to the group or where they go to another group?
So we had one case. So the one that didn't conform, Lecker, he actually moved back the week after to his origin group because he was already a secondary disperser, so it wasn't his natal group. And then he seemed to be quite confused and then started eating the pink color that he didn't conform to, but he was then very low ranked. And when he was in his new group and didn't conform, he was actually at the top of the male hierarchy because the dominant male had recently died. And so we think that maybe his different social status explained the fact that he didn't care what the others were doing, whereas all the other immigrants are usually low rank, peripheral males. And for them to be accepted by the group is very important for their survival. And so as shown in many studies in primates, homophily is really important. Behaving like others, you can bond more easily. So it might be very simple mechanisms that push them to be like others when they are not sure that they are going to be accepted in the group.
Okay. I have a last question, maybe. What do you expect between what difference can you expect between the behavior of vervets in monkeys in cities and versus in forest?
I think they have very different daily challenges. So, for example, it's very exciting to look at the alarm calling that they do in the urban areas. So they still have the snakes that are similar predators, still have eagles, but then they don't have the ground predators anymore that are in the savannah. But what they have in the urban area is many dogs. And so it's really funny to see, depending on the type of dog. So typically for small dogs, they will be doing the shutter like for a snake, and then for large dogs they will bark like for a leopard. And so it's interesting to see how they adapt this. Then also, I was very surprised to find them once at 10 a.m., all the group totally sleeping deeply, which you will never find in the wild. It's too dangerous. But then I still think we really need to put accelerometer data, because I think their nights might often not be very calm. Some of them have sleeping site on an island between two roads. And so with cars driving and people still walking their dogs and stuff. So I think it's going to be really interesting just to see the activity budget, how it differs. And then another topic I really want to look at is a lot of them eat a fair amount of human food, either from bins or stealing in houses or stealing from people at restaurants and things. And with the environmental DNA method, we'll compare the natural diet versus human diet, the percentage that they have, and then to see how little one learn what to do, in which houses to go, because in some houses it's safe and people are often not there and they leave their window open. In other houses, they have many dogs and they will never go to, or people are really aggressive to them. People are still shooting at them. So yeah, I think it's very complex, but it's amazing that they survive. And actually, where we work, it's an equi-state, and they still have a lot of natural vegetation, and they seem really to be in a lot of the time actually behaving in quite a natural way, despite being surrounded by us all the time.
Yeah. Oh, yeah. That was one of my questions. What is the relationship between the humans and the monkeys in cities? Because usually it's not. Yeah.
So I think that was really important for me, that we start this urban project, was really to tackle the human wildlife conflict that there is in South Africa and not stay only with fundamental research. And then we have a bit of both, either a monkey lover that will feed them and try to touch them, or the opposite, monkey hater, that will be very aggressive towards the monkeys. So, and both of them have to change their behavior. So we have a lot of educational work to be doing there as well.
Sure. And one very last quick question: do you expect cognitive difference between them?
So yeah, especially for physical cognition, there I really expect them to be better in the city, because they manipulate many things all the time. So pushing windows, opening doors, and things that, of course, the wild ones don't have to do. But then we'll have to see for the social cognition, their groups are smaller, they seem to be dying a lot with road accident and stuff, so maybe their social complexity and social skills are less good. So this is all what we want to try to disentangle.
Thanks Erika for this presentation. And if people want to ask questions, they can still ask you. Are you on Discord maybe?
Yes, I'm on Discord. I have now to go and get the kids out of school, but I will look later tonight and on Monday to answer all the other questions.
Thanks a lot again. Thanks a lot.